a series of 4 shorter talks, started by Professor Kadambot Siddique of the University of Western Australia, and he's going to talk about characterizing root trait variability in chickpea germplasm for adaptation to dryland environment. Professor Siddique has 10 minutes. And remember, hold your questions. We will have 15 minutes of questions and answers at the end of the talks.
Thank you, Mr. Chair. I also thank the conference organizers for giving me this opportunity. My co-authors, Dr. Yinglong Chen from University of Western Australia, Michael Gannon from ICADA. I've also acknowledged the funding bodies and other collaborators. I think we heard from the 2 previous speakers the importance of the root system. It's very critical to maintain the structural and functional properties. And greater attention is required below ground. We heard that from the day 1 onwards. I'm very happy to see that it's already happening. And we also heard from the 2 speakers the importance of the traits, deep root system. Not necessarily it's always universal for every environment. Increased root density in subsoil, increased root hair length and density. We heard the mucilaginous excretion and root hairs. And of course, we just heard about the importance of the xylem diameter in relation to conductance. So the chickpea, an important crop, drought stress do reduce yield. And the most important thing is that both at ICRISAT and ICADA, a core collection has been assembled, maintained, characterized, and there's a lot of genomic information available. So the aim of this study was to assess the detailed description. In other words, phenotyping, what is the variability available using a novel technique developed in our lab. So we didn't get the whole 300, but we used 270 and some advanced cultivars and a lot of land races. And we have developed a system, what we call the simple rubbish bin system, where the normal rubbish bin is converted into a system where there is a pump below the bin which recycles and fine jet spray of water and nutrients. So we are looking at the true genetic potential, and the paper is available in functional plant biology. By 35 to 40 days, the plants reached, at least the fastest growing reached about 1 meter, and we measured a number of both the shoot and root rates, and also we have some photographic method, and we looked at many traits such as the dry weight and density in different layers. So this is the system, so very inexpensive system. You can see the rubbish bins and the pre-germinated seeds, they're growing happily, but we were able to get some very good quantitative traits in a variety of germplasm. And I'm not going to show all the data, but the total root length is very important. You can see the variation in the Daisy collection, 207 accessions from anything about 525 to as high as 3,600 centimeters. Then the median value is shown here, and in the case of Kabuli, which had less number in the core collection, again, significant variation existed. Between Daisy and Kabuli, there is some P-shaped, which we call P-shape, and there was less numbers, but again, there was variation available. We also looked at the root diameter class, length in centimeters, and the relative diameter class, and you can see some sort of distribution system here. I think the main point is that tremendous variation exists within the core collection for that trait as well. Taproot length, it's very important because chickpeas are taprooted species, dicotyledon, and the total length again varied from just below 50 to as high as about 100, so twice different. Again, the median value is shown there, and again variation in Kabuli and variation in the P shape. Specific root length is another trait which we look at physiologically. It has got functional properties. Again, you can see the variation from 40 as high as about 250, variation in Kabuli, as well as the P shape. So whatever trait we looked, there was significant variation, some of the key parameters we tested. And this paper is already published online, and if anyone is interested, happy to provide. So the conclusion is that there is large variation in rooting patterns, and branching were observed in the co-collection we tested. 30 root traits were characterized with 17 traits having significant variation. Coefficient of variation equals or greater than 0.3. The correlation matrix showed there was a correlation among selected traits. So, for example, the root dry weight and some of the root length parameters were. The principal component analysis revealed 3 principal components capturing about 81.5% of the total variation. Okay, this is done in a semi-hydroponic system. So the next challenge, what we are doing is we're moving into the rhizoboxes with the soil and also large PVC tubes up to 1.5 meters with soil to characterize these ones and also manipulate different nutrient and water supply and studying the contrasting genotypes. The other study we have done is we have just completed phenotyping these 300 accessions for their phosphorus acquisition efficiency, phosphorus use efficiency, including this morning you heard about the role of the root hairs. So, and in addition to that, we are collaborating with Rajiv Vashni and his group, linking the genomic data. So I think our platform has allowed us to quantify the variation, then assess this variation without any restrictions, then moving into the real world, including rhizoboxes and soil, and finally into the field. So we believe that such kind of studies will help us to quantify and then breed varieties or new varieties of chickpea or other crops with root traits adapted to specific target environments. Thank you.
Thank you. That's given us a little more time for the discussion later. We won't take questions now, even though there's about 3 minutes left. But we will we'll give Professor Siddique the opportunity of answering more questions than some of the others later.